Okay, hello. My name is Jiha Kim, and I'm an associate professor in biological sciences at North Dakota State University. Our lab is interested in deciphering pathological signature of a tumor microenvironment. In particular, we are interested in vascular signature and hoping to be able to turn this around into our advantage to improve a treatment efficacy in pancreatic ductal adenocarcinoma, PDAC in short. Only about 12% of patients with the PDAC survive beyond 5 years, and it goes down even to 3% if patients are diagnosed at later stage with distant metastasis. Among many other reasons is like lack of early detection method and effective treatment options contribute most to this poor prognosis of the PDAC. Aberrant tumor blood vessels, they result in extreme hypoxia, um, which usually hinders efficient delivery of the therapeutic drugs, and also it causes all sorts of problems into a microenvironment. So to overcome this hurdle, we aim to normalize tumor vasculature into more healthy, functional tumor vasculature to first alleviate the tumor hypoxia, which, by the way, there are that knows to induce aggressiveness and metastasis, and also by having a functional normal tumor blood vessels, we are hoping to increase the drug delivery efficacy, as is the highlight in our working hypothesis. So our previous study revealed that PDAC tumor vessels are surrounded by pathological pericytes that express a significantly high amount of alpha SMA, which is alpha smooth muscle actin. As you can see here, lots of green signals on the upper panel here is the mouse model for PDAC tumor, and the lower panel here is the actual human PDAC tissue. You can see lots of the green fluorescence expressing alpha SMA expressing pericytes. Those alpha SMAs usually considered as a marker for cancer-associated fibroblast or smooth muscle cells, however, not for the pericyte. So it's clear that this phenotype is not normal. And this aberrant pericyte phenotype is also significantly correlated with the vascular leakiness. As you can see, lots of fitzy-dextran, green fluorescently labeled fitzy-dextran, are leaked out of the blood vessels. And also, it's correlated with intratumoral hypoxia, as you can see here by lots of green fluorescence here. So basically, it's correlated with lots of bad things in the tumor microenvironment. So as an attempt to identify the causes for pericyte phenotype switching or transformation, we utilize in vivo pericyte culture system. From this approach, we found out that cancer cell-derived exosomes, which are the small cell-secreted extracellular vesicles, and then a lot of those exosomes are secreted by cancer cell. So we found out that PDAC, cancer cell-derived exosomes, are capable of transforming normal pericytes into a alpha SMA expressing pericyte. As you can see here, these are the normal pericyte, these are transformed pericyte. These pericytes are also, wherever they have the alpha SMA expressing pericyte, those blood vessels were leaky. In addition, these cancer exosome treated alpha SMA expressing pericytes were very different than normal pericyte. They started to express significant amount of immunoregulatory molecules, which is definitely not the case for normal pericyte. And also, this transformed pericytes exhibited altered morphological phenotype, such as they're stiffer than normal pericyte, and they have a more like elongated shape. So definitely different, presenting different biomechanical property once they're treated by their cancer cell-derived exosome. And these phenotype are not suitable, actually, biomechanically, they are not suitable for normal pericytes and their functions. And these normal pericytes usually wraps around the endothelial tube, and that they contribute to stabilize blood vessels. And also, there's a lot of molecular communication going on between endothelial cell and pericytes normally. So, encouraged by this result, we are currently performing single cell RNA sequencing of a PDAC tumor isolated pericytes, and also, of course, comparing it to normal pericyte from the healthy pancreas. This technology will help us to have a complete molecular profile of those pathological pericytes, um, and then, that will enable us to identify a unique set of target molecules that we could potentially use toward vascular normalization approach. In addition, we are also performing this multispectral imaging for a combination of some of the immune markers and also the parasite markers. Um, to really evaluate the influence of this, different pathological parasite phenotype, and how they influence other components of tumor microenvironment, such as immune molecules. Um, and also, we are interested in understanding evolution of the tumor microenvironment as the result of the cross communication between cancer cells and tumor microenvironment components. And all these approaches will help us to have a comprehensive understanding or more of the tumor associated parasites, and also identify a normal vascular normalization approach, which will enhance the therapeutic efficacy in PDAC down the road. So with that, this project was supported partially by NIH COBRI the grant, and also NIGMS and NIH, this DaCCoTA grant. And if there's any questions, please reach out to me. Thank you.